How can a sensory cell, which extends from the toe of a giraffe to its spinal cord, quickly deliver a message? How does that message remain as potent at the end of the cell as it was at the beginning of the cell? The secret lies in the nature of that neuronal message, a sequence of changes in membrane potential called the action potential. Neuronal messages travel as changes in the relative charge across the plasma membrane. A difference in charge on the inside of the membrane relative to the outside of the membrane is called a membrane potential. Neurons have specialized features that allow them to develop a large charge difference, or action potential, that can be sent unchanged along its length. In the giraffe, this distance could be up to 15 feet. Some cells are electronically connected to other cells. Although the difference in charge is passed from one cell to the next cell, the membrane potential produced at the next cell is usually not as high as that of the first cell. Cells of the heart and smooth muscle communicate via electrical signals in this way. This type of connection works well to coordinate the functions of cardiac muscle cells, but it does not work well over long distances. Most neurons communicate information to other cells by the release of chemicals stored in packets at the end of the axon. This is how a neuronal signal travels from cell to cell. Many drugs can interfere with this communication between neurons. In this activity, we will learn how the structure of the neuron relates to its function. First, we'll explore the anatomy of a neuron. Then we'll learn how a neuron generates a neuronal message by creating an action potential. Finally, we will see how neurons communicate with one another and how some drugs affect this communication. Neurons are generally composed of three parts. The cell body contains the nucleus and organelles. Dendrites and axons branch off from the cell body. Dendrites conduct neuronal messages toward the cell body. Axons conduct neuronal messages away from the cell body. The proteins embedded in the plasma membrane play a crucial role in maintaining the strength of the message along the length of the neuron. Almost all axons and dendrites are surrounded by accessory cells. These cells create a protective sheath that also affects the speed of neuronal signaling. This sheath is made of myelin, the protein and lipids left after the cells that surround the axon eventually lose their cytoplasm during the development of the nervous system. Myelin can be destroyed by processes encountered in hereditary disorders, such as leukodystrophy, and in acquired diseases such as multiple sclerosis. The plasma membrane of a neuron contains many proteins that determine neuron function. These proteins include receptors, ion pumps, and ion channels. Special ion channels, found only in cells like neurons, play an important role in propagating the action potential. Ion channels are found all along the axons of non-myelinated neurons and at the gaps in the myelin of myelinated axons. Signals within the neuron are transmitted via action potentials down its axon. Incoming signals are received by the dendrites and the cell body. The signals may make the interior of the cell membrane more negative or more positive relative to the outside. These signals are added up, and it is the sum of these that determines whether an action potential will be sent by the target cell. Axons terminate at the synapse, the space between two neurons where communication takes place. The axon shown here is enlarged and filled with vesicles containing neurotransmitters. Neurotransmitters are the chemical signals of the nervous system. When released by an axon, they diffuse across the synapse where they encounter receptors specific to that neurotransmitter. Now that we're familiar with the anatomy of a neuron, let's see how neurons transmit their messages. Nerve and muscle cells are said to be excitable cells because, with stimulation, neurons and muscle cells have an active response. In neurons, the response is the action potential, or nerve impulse, which travels down the axon. Excitable cells have ion channels with ion gates that open or close in response to changes in membrane potential. All cells have electrically negative interiors compared to their exteriors. This charge difference can be measured by placing an electrode inside the cell and attaching it to a voltmeter. Such differences in charge represent potential energy and are called potentials. By convention, the charge of the exterior of the cell membrane is designated as 0, and the potential difference, or voltage, across the membrane of any cell indicates the negative charge of the interior of the cell. This difference in charge is due to many factors, 3 of which we will discuss here. First is the action of the sodium-potassium pump, which takes 3 sodium ions out of the cell for every 2 potassium ions it puts in. Since it pumps out more positively charged ions than it pumps in, it accounts for about -3 to 4 millivolts of the difference in voltage. Second, there are many large negatively charged proteins and nucleic acids that reside in the cytoplasm. Third, the flow of ions is controlled by ion channels in the plasma membrane. These ion channels are selective. Each allows only one kind of ion to pass through. In a cell at rest, potassium ions frequently cross through the membrane, but chloride ions and sodium ions only cross infrequently. A neuron is at rest when it is not sending a signal. The membrane potential of most resting neurons is around -70 millivolts. The resting potential of a neuron is determined by the ion concentrations on either side of the membrane. In a resting neuron, there are relatively more potassium ions inside the cell than outside, and there are relatively more sodium ions outside the cell than inside. Recall that molecules or charges will flow down their concentration gradient until they reach equilibrium. Equilibrium across a semi-permeable membrane is reached when the concentration on both sides is the same. Molecules still move back and forth across the membrane, but there is no net flow in any one direction. The differences in concentration gradients will tend to force potassium ions out of the cell and sodium ions into the cell if these ions can find an open path through the membrane. The potential across a membrane can change in response to a stimulus such as physical contact, electric charge, pH changes, or ion flow. The axons of neurons contain sodium ion channels that open when a particular charge difference, called the threshold potential, is reached. When the gates of the gated sodium ion channels open, sodium ions rush into the cell, and the internal sodium ion concentration approaches that outside the cell. Then the ion gates close again. Although the opening of the gates dramatically changes the membrane potential, it happens for only a millisecond or so. This opening and closing of the gated ion channels produces an action potential. Because gates for a particular ion are either all open or all closed, an action potential is an all or none response. Ion channels that open and close in response to certain membrane potentials are called voltage-gated channels. After voltage-gated channels open and close, there is a time period, lasting about 1 millisecond, when they will not open no matter how strong a stimulus is. This period is called the refractory period. This assures that the action potential travels in only one direction, away from the cell body where it originated. Let's follow the changes in charge that take place along one section of the axon membrane during an action potential. A neuron receives a stimulus that causes the sodium ion channels to open. Because there are a lot more sodium ions outside the membrane than inside, sodium ions rush in. Remember that the sodium ions have a positive charge, so the interior of the axon becomes more positive. The charge differential will be restored to the resting potential by the opening of voltage-gated potassium channels that are triggered by the same stimulus that opened the sodium channels. The potassium channels take longer to open than the sodium channels. When they finally do open, potassium ions rush out of the axon and down the potassium concentration gradient. This makes the interior of the axon more negative, reversing the charge difference. Also at about this time, the sodium channels close. Gradually, the ion concentrations go back to resting levels due to the action of the sodium-potassium pump. In myelinated neurons, voltage-gated sodium channels are located only at the nodes. When the gates at one node open, they produce a current great enough to get the next node beyond its threshold potential, thus passing the signal on. This leaping action potential is called saltatory conduction, derived from a Latin word meaning to jump. Once the action potential reaches the end of an axon, its message must be transmitted to other nerve cells. We'll see how in the next section. Axons terminate at the synapse, the space between the neuron and its target cell. Here an action potential causes voltage-gated calcium ion channels to open. The resulting influx of calcium ions causes vesicles containing neurotransmitters to fuse with the synaptic membrane, releasing the neurotransmitters. Neurotransmitters diffuse across the narrow synaptic space and bind to receptors on the target cell. These receptors are ion gates that are ligand-controlled. When bound to their specific neurotransmitter ligand, the gates open, allowing the flow of ions, either sodium, potassium, chloride, or calcium, into the target cell. The change in ion flow causes a change in the potential across the target cell's membrane. In order to cause an action potential in a neuron, the sum of the potentials induced by the binding of neurotransmitters must reach the threshold potential of the sodium gates in the region of the cell nearest the axon. Click on the synapses to see what combination of stimuli will induce an action potential in our target cell. You can watch the screen to see how the binding of neurotransmitters at each synapse affects the charge across the target cell's membrane. Neurotransmitters tend to be small molecules. Around 30 different types of neurotransmitters have been identified. They include the chemicals carbon monoxide and nitric oxide, as well as amino acid-based molecules like acetylcholine and norepinephrine. A deficiency of the neurotransmitter dopamine is seen in Parkinson's disease as cells that produce dopamine die. This disease is marked by tremors, rigidity, and an unstable posture. The drug L-Dopa is chemically related to dopamine and can lessen some of the symptoms, but it cannot reverse the eventual progression of the disease. The action of the neurotransmitters lasts about 1 millisecond. The mode of inactivation differs depending on the type of neurotransmitter. Some simply diffuse away from the receptor. Some are digested by specific enzymes. Others are taken back up by the neuron that released them and recycled for later release. The nerve gas serin works by inhibiting the enzyme that digests the neurotransmitter acetylcholine. This leads to continuous stimulation by acetylcholine, which results in paralysis and respiratory failure. Most drugs that can alter the mental state exert their action at the synapse. These drugs either promote or decrease the action of a particular neurotransmitter by blocking its action, mimicking its action, or preventing the termination of its action. For instance, cocaine prevents the reuptake of dopamine by the axon that released it. The result is a longer period of stimulation by dopamine which induces euphoria. This extended dopamine action has many serious consequences. First, it is highly addictive. Additionally, the continuous presence of dopamine causes the neurons to respond to the excess by making less dopamine. Thus, there will be a deficiency in dopamine response, producing feelings of fatigue and mood depression unless the drug is present to artificially raise the dopamine level again. The differences in ion distribution on either side of the membrane results in a voltage difference, potential, across the membranes of all cells. The sodium-potassium pumps and the differential permeabilities of ion channels in the membrane create a concentration of sodium ions that is higher on the outside of the membrane and a concentration of potassium ions that is higher on the inside of the membrane. Neurons may respond to changes in the potential across their cell membrane by firing off an action potential. Incoming signals from the dendrites and on the cell body produce ion flow changes that add to or subtract from the cell's resting potential. If the sum of the changes reaches a particular voltage, the threshold potential, an action potential will be triggered. A signal is transmitted by the propagation of action potentials down the axon. The axon contains sodium channels that open when a particular voltage, the threshold potential, is reached or exceeded. When the gates are open, sodium ions rush in and the interior of the cell near the sodium gates becomes more positively charged. The voltage-gated sodium ion channels in a particular part of the axon will close after a brief time, but the voltage-gated sodium ion channels of the next part of the axon will be stimulated to open because the local potential now exceeds the threshold potential. The opening and closing of the voltage-gated sodium ion channels results in the difference in charge called an action potential. Action potentials are all or none responses since the gates are either open or closed. The charge difference is returned to the resting potential when voltage-gated potassium ion channels finally open. Potassium ions rush out of the cell, down the concentration gradient, reversing the charge difference across the membrane. At about the same time, the voltage-gated sodium ion channels close. In the refractory period, the voltage-gated sodium ion channels will not open no matter what the stimulus strength may be. This assures that the action potential travels in one direction, away from the cell body where it originated. At the axon terminal, the action potential causes voltage-gated calcium channels to open, which leads to release of neurotransmitters from storage vesicles. Neurotransmitters diffuse across the synapse and bind to receptors on the postsynaptic neuron. These receptors are ligand-controlled gates which change the flow of ions in the postsynaptic cell. The change in ion flow causes a change in the potential across the membrane, either closer to or away from the threshold potential, depending on the ion. Neurotransmitter action can end as a result of diffusion, digestion, or reuptake. Nerve gases and many drugs that alter mental states exert their action by altering neurotransmitter function, release, or uptake. Although the mechanism by which a neuron sends a signal is well understood, we are still learning about how the coordinated firing of individual neurons is the basis for our ability to move, breathe, and even learn.